Hi. So now the next topic is about how to remove the intron to make a final mature messenger RNA. Although it is a very complicated procedure, and then actually, this is the procedure in which actually the original discovery of ribozyme is made. So, um, actually, this RNA providing this catalytic activity of this splicing of a particular area of the RNA was discovered, and now hence the ribozyme, the name was coined. But anyway, we are not going to look at the detailed complex mechanism, but simply zip through, and then from the actual utility aspect of why, then what's the significance of this um, the RNA intron and splicing, RNA splicing. So the intron, why they are called intron is originally after realizing there are certain segments that are not actually being used in the final version of the messenger RNA, because after all, if you think about this utility of messenger RNA, they are going to be used for protein coding, and even originally some area segments are present in the part of this coding region of the messenger RNA, and they are not going to be used at all in the end. So they are interrupted. So in other words, after removing this intron segment, and then you do this splicing job of the remaining exons only, then now you get to realize that, oh, those were the something that's kind of originally acting as an interrupter of this our real significant in terms of the protein coding activity, significant on exons. So that's why they were called introns. And so this RNA splicing is one of the three major the messenger RNA processing in the eukaryote. So here, actually, the other two, the steps or jobs of RNA processing are also described in this figure, although we are not going to mind at all, focus on any of this attention on the other two. But so here, the introns are there and exons were there in the original primary, or sometimes it's called the pre-mRNA, and all these introns are gone, and so they in the mature messenger RNA only the exons for protein coding region they consist of. But additionally, what you also notice hopefully is originally in here, both at 5' end and 3' end, you didn't have this segment. Whatever they are, their names weird. The 3' end is called the tail. Fine, tail, because the 3' end is anyway, after all, is a tail, and it's called the poly A tail. It's because there are some meaningless repeat of A, just like that. Something like was like 20 to 200, something more than 200 repeat, and that's not something that originally present over here in your coding the DNA, but later addition it was being added from somewhere else and then added into some kind of plug. And you do have another plug at the other end, at the 5' end, named as a 5' cap, like baseball cap, another plug. And both of them are actually acting as a protective roles and enhancing the protein synthesis efficiency. But we are not going to take a look at any of those, but simply. So These, if you just wondered what are those three major RNA processing in eukaryotic messenger RNA, does that mean you do not see any of these three different processing in the prokaryotic messenger RNA? Yes, answer is simple answer is yes. Although in the archaebacteria is a different story. So that's why we can actually have this standard form that we are more close. I shouldn't say we are closer relative to this archaebacteria than the real bacteria, the present day real bacteria we call the eubacteria. But anyway, so they do have these introns and then the features just like we do. So these introns are so they are removed, as I mentioned briefly in the previous lecture session. Why do we still keep this? Because they're not going to be used. So actually, there are two different, sharply divided, two opposite schools in the academia in the explanation of the introns presence, why they are still there. Sharply divided in theories. Oh, because of this, it's there. And origin and then theories. But it's a still controversial debate, ongoing debate, so we won't even touch that. More importantly, if we get to think about this more practical aspect of introns presence, is there something really something that are garbage, obsolete one, or are there any kind of the second-hand usages so that we can make use of this under the circumstances? It appears that the latter is the case. It's so even if not every genes, but some genes seems to be able to develop or evolve the how do you utilize this intron presence in to their benefit, so that bottom line is cutting all those crap out. The bottom line is by having this different intron area, one can enjoy the luxury of generating more than several different versions of a protein out of a single gene is the concept, and then that's the reality. By alternatively, okay, this is on Monday, for example. This is Monday, so it's your turn to become an intron. Oh, tomorrow is a Tuesday, so it's your turn to become an intron. Just like you take turns. To give you an example, of course, they don't take turns and by the different days, but obviously they take some turns where you are, at least by different tissues in your body, or in some other occasions by like developmentally, so differentially, in the early developmental stages, your certain area becomes intron and the other alternatively area become axon and etc. So this is another example of if you look at closely, if you try to examine and make yourself understood the sense out of this thing, is this is one real life example of a primary transcript of any genes. So here the blue bar represent introns, so you have several introns. And there are several different versions of exons. Okay. But now what you see is you maintain two alternative variants out of this primary transcript. Once you make the RNA, that's the primary transcript, isn't it? Or pre-RNA, pre-messenger RNA. Then now it's time for this RNA's the intron splicing. So you get rid of the intron and then exon splicing. That's what you are going to do. But in that, you alternatively choose different segment as the axon, final axon. So if you are the one actually unfortunate area that finally not to be chosen, then you are technically just just another intron, isn't it? Even if here originally you were labeled as 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 segment of exons, but if you are not included in the final version, then at least for the day you are not the exon, but the intron. And this is something called the alternative splicing. So here, two different variants, two different versions are employed. So then one version goes to the cell membrane and then become as a cell surface membrane protein. Once you generate this type of version, then this protein goes to the cell membrane and then promote so cell death of this particular cell. There is some particular procedure called cell suicide, cell death. Unfortunately, we don't get to reach to that far in this semester. But on the other hand, somehow the alternative version, just you check yourself. What's the difference between these two? Even though they are from the same gene and same primary transcript, but two alternative versions are generated by different splicing. So here 1, 2, 3, 4, 5, 6, 7, 8, 9 are all these blocks are present, but here block number 6 is deliberately omitted. Then now you have 1, 2, 3, 4, 5, 7, 8, 9. Then now you are not able to, the final product as a protein, you're not able to go to the cell membrane, and this version will stop this process of a cell death. So two quite distinct opposite outcome can be controlled by the single gene. Although this is very straightforward example, but in real life example, there are more actually bizarre and complicated example of operating several different versions of this alternatively spliced version out of a single transcript, and then you control your cellular metabolism like that. So, hey. So this is called alternative splicing. And striking evidence for the broad occurrence of this practice of alternative splicing is when we recognize after the Human Genome Project completion, because even before the onset of the Human Genome Project, scientists knew that there are at least 100,000 different proteins present in the human cell, any given human cell. How many different proteins are present? Let's find out. And then they were able to figure it out. So then, hey, the numbers are 100,000. So then everybody can expect, okay, if the protein is 100,000, then what's the minimum number of genes present in our human genome? Obviously, even if you are not a rocket scientist, then you can easily answer that, because it's got to be at least more than 100,000, because each gene will be responsible for making one protein, and especially if you consider there will be some other non-coding genes. Only some of the genes will be responsible for directly coding the protein, the messenger RNA gene, but there are also ribosomal RNA gene, transport RNA genes, and etc., non-coding genes. So then, obviously, you can say with confidence that there's got to be at least 100,000 different genes present in our human genome, but it turned out that when it's all said and done, or when it is completed, then the number of the different genes in our human genome was only merely about 22,000. What is this? What is going on? It's like even less than 1/4 of different number of proteins. How can it be possible? How can you generate this more than one different version of the protein? You have only so many genes, like only 22. So the only way possible is through this alternative splicing, so that one gene can generate two or three different genes, or even more than four different genes, then can it be only possible, right? So that's the aspect of this alternative splicing and the utility. So for that matter, introns, after all, the keeping intron may not be such a stupid idea, so that you can operate such a the higher level of this more exotic, so to speak, at least exotic gene regulation and maintenance, just like we demonstrate. And this is not only the case of the human or an animal. Everywhere in eukaryotic the world, in plants and in fungus and everywhere in this messenger RNA genes carrying this intron, then you get to observe in these days, oh, you guys are just maintaining, operating this alternative splicing just like norm. Okay, so that's the probably final message about those gene expression aspects. So now we can jump into the next topic of actual information conversion procedure, actually making some tangible protein out of this information. So this is overall called the central dogma, describing the direction of the information flow from DNA and RNA in protein. The significance of this central dogma looks like what it states directly is the genetic information only flows from direction over from DNA to RNA to protein. And the more importantly, more subtle but more important message over here is you cannot go back. You cannot generate the RNA starting from protein, and nor can you do the same thing out of RNA to DNA. Okay, so that's the more important message of this so-called central dogma. However, this central dogma, the general universality of central dogma, has been beaten badly by the presence of a certain type of virus. Certain type of virus actually can do this. What central dogma? They can generate DNA, double-stranded DNA, starting from their RNA, because this virus genome is made of RNA. But this RNA genome is something somewhat different from this famous, in these days more famous COVID virus genome RNA, entirely made of RNA. But this virus is called the retrovirus. Because they generate their DNA retroactively. So starting from RNA, they generate DNA, which is DNA, the double-stranded DNA, right? RNA is a single-stranded. And then this DNA can be integrated in the host genome as part of, so if this is our host genome and there the RNA is being inserted into somewhere, like this. So this retrovirus, there are several different kinds of retroviruses do exist in that infect human, and probably one of the most famous such the retrovirus is HIV virus, human immunodeficiency deficiency virus, which cause the disease AIDS, the immunodeficiency syndrome. So this is one such good example of a retrovirus. But anyway, those weird viruses can overcome this limit, so-called limit of whatever you call the central dogma, because they have their own very unique enzymes which allows to this unusual conversion. However, later, as our level of study is progressed, enhanced, then nowadays we realize that actually this this particular enzyme capable of converting RNA into DNA is not such a thing that only limited in the virus genome, but we actually, human and plants and other organisms also have such enzymes. So that's what we have found later on. So anyway, this information, according to this central dogma, the information flow, so the actual genetic information present in the DNA will finally delivered into some type of organization, so that the protein building process, in that process, that information is used to reflect. Okay? So how? The problem is then you have a DNA consists of 4 different alternative choices of this each, because there are 4 different nucleotides present in the DNA. And there are 20 different amino acids. So then are you saying that this using this by juggling around these 4 different kinds of nucleotides, then you are going to specify what I mean by that. You are going to specify in whatever the form that your information present in your DNA to specifically to each of 20 different amino acids. Remind you, in protein building, there are 20 different kinds of amino acids are used. So basically what we are seeing is that although many such 100,000 different human proteins are just a result of this different combination of the orders and combination of each different 20 different amino acids, then can you generate so many different variety of the proteins. And if you think about that, the mathematically the potential has not at all been reached yet. So we do have so many more potential of being able to generate many different versions of the protein, because we have this 20 different amino acids. Okay. And anyway, so here this the relationship is set by this 3 nucleotide unit. So the codon that is referred to as codon, and what codon is the set of 3 nucleotide which we can find on the messenger RNA. However, since messenger RNA sequence is directly complementary to the template DNA sequence, and then that template DNA sequence is also complementary to the complementary sequence of the DNA, so we exactly can find the exact same codon in the DNA, right? So that's the beauty of this complementary base pairing relationship. So if you want to know about the status of any specific RNA, you look up. You do not have to go further anywhere else, but simply take a look at your DNA, and you can easily find, without even any, there's such a simple conversion business, but directly you will be able to see exact the nature of a codon in the other strand of your DNA. That's why in the previous lecture I mentioned that the non-template DNA strand as what? Sense strand. Because if you look up the non-template DNA sequence, then it's much easier to figure out actually what kind of codons could be generated by this area of the gene, right? So this codon, single codon, consists of 3 nucleotides. Why specifically 3? To explain is mathematically speaking, if you have decided to use 3 as a one single unit, out of you have 4 different nucleotides, and then how many different possibility of, if you decide this is 3 nucleotides in a single codon, how many different codon can you generate? It's 64, isn't it? Because you have 3 positions, the first, second, and 3, the third position. Each position will be occupied by one specific nucleotide out of 4. So how many different cases can you think of? There are 4 different cases. Out of 4, how many different nucleotides can you pick for the first codon 4? And how many different codon can you find for the second position 4? So 4 by 4 by the third 4. So it's 4 to the third, which is 64, right? But, 64. You have, what you need is only 20. Why do you need any 64? This is gross waste. But if you think about it, so this is just the weight, so let's just cut it down. Let's slim down our thing, economize. But then you soon immediately realize that you are not able to cover even enough these 20 amino acids differences. If you go down just by 1, so that once you decide to use only 2 nucleotides, then now the different possibility of generating different codon is suddenly reduced into 16. So that you cannot function. So anyway, so each different codon which correspond to specific amino acid, and usually by convention, specific different amino acids is described in 3 letters. Okay. So it's so like 1, 2, 3, A, U, G, 3 different nucleotide on the messenger RNA specify one particular amino acid, methionine, but usually conveniently we just abbreviate it into 3 letters of methionine. But actually, an expert, not only in this field, these biologists and chemists usually even go by 1 letter convention of, like, doesn't even bother even using 3 letters. Just 1 single letter. M is another way of describing this methionine amino acid. On the other hand, G, 3 consecutive appearance of this nucleotide guanine signify another amino acid, glycine. In this case, the single letter G can alternatively be used. In the case of a 3C, consecutive C, code for proline in here, guess what? It's P, single letter P can take the place of 3 letter proline, and it just go like this. So all these 3 different and 20 different amino acids, there will be some kind of different specific order of 3 nucleotides as codon exist, is the point. So that here the relationship goes like this. Overview is, in the sense DNA ACT, 3 specific ACT nucleotide which can be complicated, complementary to the TGA template DNA, which is complementary to ACU messenger RNA. And then if you go now compare this messenger RNA codon ACU and the sense DNA, which is non-template DNA strand, ACT versus ACU, so the only difference is a T to U conversion. In the case of next codon, which is a CCU, and once again CCT, so T to U conversion. And GAG, which is another codon on the messenger RNA. Then now if you look at the sense DNA version, GAG, same, exactly identical. That's why this non-template DNA sequence is called sense DNA, because it makes much more sense in the case of a codon identity. So this, once that messenger RNA generated is finished, it's completed, then this messenger RNA can move out of nucleus. Then now it can be engaged in actual protein synthesis procedure. And now each codon will be matched to specific amino acid. Now here the question is how how each different messenger RNA can specifically match into specific amino acid. So the secret is now via another establishment of complementary base pairing between messenger RNA and now in this case the transfer codon. Because here what you see is codon and there is a something called anticodon is there, and the codon versus anticodon relationship, what you see over here is complementary. And where can you find this anticodon sequence is the question. Guess what? It's on the transfer RNA. What's the function of the transfer RNA? They transport each different amino acid. So now, if you can somehow link this relationship, okay, if transfer RNA, each different transfer RNA can carry a specific amino acid, and each transfer RNA carry their respective very specific complementary base pair area we named the anticodon, then now it's just like transfer. Each transfer RNA can function as a kind of adapter. So between messenger RNA plug and maybe messenger RNA is the plug and transfer RNA is a code. Then you just plug in. Then now this transfer RNA has some specific amino acid is there. That's the relationship exists between messenger RNA and each specific transfer, the amino acid. Now, having said that, then how many different transfer RNA do you have to have in your cell? At least you should have 20 different transfer RNA, each of which they can recognize and bind and carry different kind of amino acid, right? However, if you also consider this, how many actually different codons are possible? We have just looked at, there are 64 different codons that are available in our cell. So in order to actually cover the entire 64 different possible codons, actually the number of the anticodon sequence, the number of different transfer RNA carrying different type of anticodon, each of which matches with specifically to specific codon via the complementary base pairing, then you have to have 64 different transfer RNA. So now there is some kind of a confusion discrepancies. Transfer RNAs carries the amino acid. There are only 20 different amino acids. And now the number of transfer RNA that can match to your all the possible codon must be 64. Then how do you manage? Multi relationship in the transfer RNA if you allow. So that's what it is. So 20 different amino acids used in protein. So the codons, because out of 64 codons, if you are going to assign 20 different amino acids in this fashion, then obviously there's no way but you actually have to allow this redundancy. In other words, for one single amino acid, more than one codon should be allowed to specify. That's what it's called, codon redundancy. And this one is just like the thing that I have just mentioned. This codon, if those codons were made 2 nucleotides, then you can cover only 16 amino acids, so that is not enough. On the other hand, if you would not be that crazy to have like more than enough 4 nucleotides in one single codon, then now all of a sudden the different possibility becomes increased up to 256. So that's just totally unnecessary, right? So that's why this 64, even though this 64 on the surface apparently may seem unnecessarily more than enough, but in reality it is just exactly what you have to carry. Otherwise you're in trouble. So each of these three nucleotide on the codon and then the messenger RNA is now recognized and then can be bound specifically through this complementary base pairing by each different tRNA. So those specific three nucleotide which is complementary to the corresponding codon is called anticodon. So between codon and anticodon, here, just like that. So this anticodon sequence in reality, forget about this amino acid interfering in between. No, that is not actually just the case in reality, but directly there will be some interaction between this codon and anticodon in the form of transfer RNA having this anticodon sequence exposed so that they can have this binding. And where is the amino acid? The other side. Amino acid on this particular transfer RNA is the other side, somewhere like this. That's how it is in reality, just like this. So this is one such molecular model of transfer RNA structure. And at the one end, at the base, actually, the 3 nucleotide is exposed, which is capable of having this complementary base pairing. And the other end here, at the other end of this transfer RNA, you have this particular amino acid is attached. In this particular case, one such amino acid, the named tryptophan. By the way, so what's the one letter the specification of this tryptophan instead of a T? No, in this case, W is the letter the representing this tryptophan amino acid. You don't have to, of course, worry about and memorizing such things. So this transfer RNA carries a single anticodon, and each different transfer RNA carry the specific amino acid, but 20 amino acids. So more than one different transfer RNA will be able to pick up the same amino acid. By doing so, you can generate this so-called redundancy. The other thing about this transfer RNA's anticodon and codon is, it's weird enough, even though there are theoretically 64 different codons, and then all of these 64 different codons are realistically generated during the course of this gene expression. Yes, it's there. However, actually the transfer RNA that matches with all those different variety of codon sequence, there are only 61 transfer RNA having different anticodons. So 3 are missing. And actually those missing 3 are the blessing in disguise. So we'll get to that. So that these 3 missing anticodon, which is not available, the counterpart, will be able to be used as some other specific way during the protein synthesis business. So anyway, here already you can have this clue. So these are the summary of all those combination of 64 different codons, and each of which particular amino acids are matched through this relationship. And once you notice these 3 codon sequence, UAA, UAG, UGA, 3 different codons, they are called the stop codon because they don't have any corresponding amino acid or anticodon. So once the protein synthesis machinery reach it here, then those machinery will stop. It's not able to make any further progress, and it stalls there. So that's how you can end That's how you can finish the process of protein synthesis. So in that way, this so-called stop codons, just like it's a stop sign. So now you gotta stop and then go home kind of a sign is this. So this is the cartoon illustrating the initial stage of this protein synthesis. Once again, this whole procedure is very complicated business. So, but luckily in this class, we don't really have to go into that such miscellaneous trivials. But simply put, each different tRNA, forget about this ATP whatever, an enzyme whatever, each different tRNA will be able to pick up particular amino acid, right? There are only 20 different amino acids that can be part of this synthesis. It does not say that there are only 20 different amino acids in the whole world. Yeah, there are probably more than 100 and 200 different amino acids available in the real world. But for some reason in the cell, the particular type of kinds of amino acids that are utilized and to become component of the protein in the living cells are only 20. We don't know why. And we don't know why. Well, probably we can explain why. There are only certain number of transfer RNA that can recognize a specific type of amino acid. That's the kind of limitation. If we can somehow create artificially a different type of transfer RNA having different types of anticodon, then that means we can generate more different type of new amino acid that get inserted into the protein. That's kind of the math scientists like the attempt, but actually it's been already made by some group of scientists, such artificial protein synthesis. So anyway, and then what you have now is you just this. The third thing that you need to understand is the actual site of the protein synthesis. What I mean by the protein synthesis? It is the procedure by which individual amino acids are connected through a specific type of covalent bond. Do you want to know the name? You don't have to remember the know for this class, but I will just give you the number. It's called peptide bond. This is a specific type of a covalent bond between two amino acids. Yes. And by simply connecting more and more peptide bond along with all those different amino acids, just like if the amino acids are individual beads, then now you are making a long chain of a necklace made of beads is the initial product of the protein. So this peptide bond formation is made in the ribosome. So this ribosome, you recognize there are two different parts, large subunit and small subunit, whatever, but they are the site of this the synthesis, means the site in which these peptide bonds are made. So through this peptide bond, two amino acid. So here what you can figure out is there are two spaces available for each transfer RNA carrying this amino acid hooked up can enter here. In other words, these two amino acids can meet in the very close proximity by way of this binding of these two transfer RNA. What's the basis of these two particular transfer RNA get in the proximity, which will be totally dictated by what? This. What kind of a messenger RNA codon sequence is there will determine what type of a transfer RNA can come so that they can connect through the this complementary base pairing. Then once you have this two spaces are occupied by these two amino acid having this like that, then now this connection, bond formation will take place. Now this figure describes the this peptide bond formation in the middle of this protein synthesis business. So already one transfer RNA now carries not only one but 1, 2, 3, 4 already, 4 different amino acids connected on the head of this transfer RNA as a result of such previous incidences of the peptide bond formation between these two. So this was the one on your left on the slide is the older transfer RNA, and then the one that on your right is the newcomer. And this green arrow, take note of this green arrow. Always this transfer of amino acids through this peptide bond formation is made from the older transfer RNA to the new one. Why? Because the older one, the fate of this older transfer RNA carrying this already made existing amino acid chain will soon be kicked out. You go out. It will be evicted, so that in order to make room for new amino acid carrying transfer RNA can enter this. That means this ribosome will migrate, will move this direction, one by one, by 3 nucleotides at a step. So by doing so, you just get automatically kick the previous one out and welcome in an open arm for another new transfer RNA having the new amino acid, right? So if you don't take care of this conversion from the older one to the new one, then you are in trouble. You will lose all those previous work of art of this protein synthesis business. So that's how you have to. And here what describes here in this actual figure is how it ends. So do you see over here stop codon, which means so you keep moving one by one like this, so you somehow achieve this transfer and moving, so you build up certain lengths of this amino acid as a one type of a protein. Now what? And then finally you have reached the area where stop codon in combination, stop codon is present. What did I say about this stop codon? There is no corresponding anticodon for stop codon. So no transfer RNA will come to bind to this region. No matter how long you wait, you keep waiting, so nothing will happen. Or actually, in real life business, this ribosome, whole ribosome complex, doesn't have any such a patience. Very quickly, is very hot-tempered, impatient person. So if there isn't any new tRNA occupies this open empty space within very short period of time, then this ribosome will discharge. Okay, I have enough. I will go home. And that's the end. That's how you now you release this made polypeptide. It's called the polypeptide. So I mentioned already the polypeptide is synonymous. In most of the cases, polypeptide is synonymous to protein. Why? Because this polypeptide means something that builds through this multiple peptide bonds is what it means, polypeptides. So in most of the cases, actually, single polypeptide equals to single protein. However, some other occasions, like hemoglobin, right, 2 alpha and 2 beta subunits all together generate 1 hemoglobin. So in this occasion, you have 4 different polypeptides to make 1 functional protein. Is in some of the occasions that you have to use such a way of this description. So anyway, this is how you have this translation termination. By the way, what do you call this business of protein making? So if we call this gene expression by which the RNA is being copied as a what? Transcription. Then now, this from the RNA messenger RNA message to build this real protein is called translation. That is very intuitive and reasonable enough. So transcript is something kind of copying, just like in the United States is when if you go to the university registrar's office and then ask for the copy of your the academic record, your grade point average, and then the 성적, then you ask for is can I have my transcript? Yeah, that's how they call. So this transcript is just like meaning the copy. Now here, the reason why it can be called as a translation is you can understand easily. So now this message is although how much valuable that may be, but it is simply just a message, information itself. But if you can somehow convert this message into something real, actual event of protein synthesis, now you can call this as a translation, because this ribosome can read the message encrypted in the messenger RNA in the form of a codon, and then this ribosome somehow happily can somehow convert into real protein as well. So these 3 stop codons and stop codon business, that's what it is. Okay, so before moving into the next section, which is about mutation, which is our final section of this chapter, I will probably I am compelled to stop over here and then wait for another session to finish this the third section.